really dark.
Is there a PowerPoint that goes along with this methods lecture?
Is there a PowerPoint that goes along with this lecture?
Yeah.
No, I have a few scattered, but it's not,
Okay.
not the way the others were. Sorry for my foolish attempts to get this done. So today's lecture is going to be on some methods, and I've been conflicted as far as when I want to teach this lecture and how much I want to put in, and et cetera. But I think it's appropriate, and we're kind of putting it in here. Well, what are we, fifth week, I guess. There we go, don't mind me. Okay, here's our stuff. So there's a bunch of links here, and one of them has a couple PowerPoints from an old Kubi, but not the whole lecture as before. So I think this is almost a discussion of philosophy of science, but I think often what we learn is technology allows us to make steps forward. I think of DNA sequencing, for instance. And I think in immunology, that's another example where we think of something like flow cytometry. We can't really conceive of the science as we know it without developments in the technique of flow cytometry. So I'm going to talk about that a bit today, but I'm going to start with something different and talk a little bit about mouse strains. Start with something just a little bit. And I thought it was going to be clever. When I originally wrote a version of this for another class, I wrote Of Mice and Men, which is a John Steinbeck book. and then I thought, oh well, I should say, I cut out the of, and I should say men and women. So anyway, started out as me trying to be clever, and ended up not as good as one might have thought. So some things. Now I know some of you have worked with mice and may know more about mice than I do. One thing to note, whereas many systems are used in biology, you probably never made a point of writing it down, but in general biology classes in genetics, you probably worked somewhat with, well, let's see, E. coli, or had presented to you experiments originally done with E. coli, probably yeast, the fruit fly, Drosophila melanogaster. Some of you have been exposed to C. elegans lately, some more than others. In the past, frogs were used a lot for certain things. Well, immunology is unusual in that almost all the work has been done in two species. Again, in humans and in mice. And, for better or worse. Occasionally you can find things done in other species, but largely in those two. And I don't know if you ever reflected on that. And not only that, the kinds of experiments that are done are typically different. So in humans, in general, we don't do prospective experiments. A lot of looking back at the past, like seeing, if people were vaccinated in the past, what's their immune response now? I guess that's not really correct what I said. I can't do prospective experiments because you can have controls and control groups and people who will receive a vaccine, but you get the idea. Humans are outbred, which sounds like a weird thing to say, but contrast that with the mice being inbred. So everybody in this room, we differ a lot at MHC loci, so we're very different. And that's kind of more of that in the next lecture, but there is great variation in the MHC genes in humans. So that's incredible. I've never been entirely comfortable with the explanation for that. Sure, it's beneficial to the population, to the species as a whole, if there's great variation with different antigens, but selection acts at the level of the individual, so it's always been a little bit of a puzzler for me. But anyway, great variation there. So the MHC, this is Wednesday's lecture, many genes and many, many alleles at each locus. In fact, especially for humans, there's a lot of lore about fine points and trivia and interesting things about human HLA, which is the human version of MHC locus. And of course, this is very practical because of experiments like bone marrow transplants, where you want to match things up. So people needed the ability to distinguish MHC in different individuals. And there's a tremendous amount of work there, and a lot of the early stuff here in Seattle at Fred Hutchinson. Okay. So the kinds of experiments, I guess, maybe not the basic science experiments so much, but clinical things done in humans. People typically sample blood. Someone can do that, take a sample of blood from people and see, for instance, level of white blood cells. High levels indicate an infection. Low levels of white blood cells might be a problem with a genetic deficiency. And of course, we can look for antibodies in the blood. In contrast with mice, it's kind of a different story, and a lot of cool things have been done with mice. Some people who were very, very clever, and I really, sometimes in science, you think about things that people started or kinds of experiments that are just very, very clever and very, very important, that lay the foundation for lots of stuff. And those of you working at C. elegans probably respect Brenner for developing that as a system. But in the early 20th century, some of the people started inbreeding mice. So they were only crossed with their own strain, and thus they were able to generate a bunch of different strains that have been inbred for many generations and should be homozygous at all loci. I guess, I don't know how they worry about the occasional random mutation, but anyway, these mice are essentially homozygous at all loci, including the MHC. And this makes experiments cleaner, if you will, because MHC is so important in an immune response, and you know what you're working with. Okay? Now, the criticism can be made, this is also a little bit contrived, so with these, but with these experiments. But anyway, know what kind of work has led to this. Okay, so people done these experiments, and a common thing is to treat a mouse in some way and then harvest lymphocytes from the spleen or lymph nodes at some point later. Cross within the strain of the same MHC. What kind of experiments? So people can mate mice at will. So if they have there are mice of two haplotypes. Do you guys know the term haplotypes from genetics? Okay, maybe a little, maybe not, maybe not. Okay, so if you have a bunch of, so here's a region of DNA, and on one strand, I'm just going to say you have these genes are arbitrarily distanced apart, and let's just say big A is in the same piece of DNA as big B and the same as big C, we can follow the inheritance of these together. And over a small number of generations, which when we're talking about humans, small might be 25. If these are close together, there's not likely to be a crossover between these, okay? And then, so you're gonna have big A with big B and big C, okay? And we speak of that as a haplotype. You know what haploid means. Similarly, little a, little b, and little c if they're all on the other chromosome. Okay, they'll be inherited together. So this you hear haplotypes sometimes when people are searching for an allele that causes a disease, because let's just say the allele that causes a disease is in some unknown gene here, and then people can follow that haplotype through many generations. And there's unlikely to be a crossover within that over a short period of time. Okay, so that's haplotype in general. In particular, when we look at the MHC locus, genes tend to be inherited together, and there's something special going on. It's even more, recombination is suppressed. So it's not just that it's one locus, and there's even less recombination than you might think. Anyway, more on that Wednesday. So the different mouse strains have a given haplotype, and that means certain set of alleles, and it also means that often if you know one allele at a certain locus, then another locus nearby will have a certain allele. So you know what you have, and then also you can do experiments only on that one strain of mice with a certain haplotype, or you can cross one strain with another with different haplotypes, and you know what you have. Okay, so you can mate mice at will. I know there's huge temptation to make jokes about that compared to people, but you can mate mice at will, harvest their organs. You can also do adoptive transfer, transferring part of the immune system from one mouse to another. And you can do that with the same strain, so there's not a response just because of the mismatch of MHC. So you can look at a particular aspect of the immune system by transferring from one mouse to another, say after giving an antigen to one. You can irradiate mice to destroy the immune system and then add back immune system from others. You can remove the thymus, add a different thymus, so you get the idea. You can transfer only B cells from one mouse to another mouse of the same strain, or a particular clone of T cells from one mouse to another. So all these kinds of things you can do. So one can be creative in that respect. Okay. And you'll see some of the experiments we'll talk about depend upon this ability to do this. Yeah, so that's called adoptive transfer. And the kind of experiment one can do is in a mouse that has an autoimmune disease, you can transfer only T cells from one strain of it. So does that transfer the disease? Those are the kinds of things, you get the idea. So there is this great history, this lore associated with mouse genetics. And these guys have been doing this going back, one name you hear is Snell, back to the early 20th century. So here are two, I'm not a mouse person, so I don't know that much, but here are two strains that have been used the most in immunology and probably the most in other areas as well. That's a C57 black 6 or B6 mice, and also BALB-C mice. So, let's see. Let me go to that in a minute. So I think this is from an old edition of Kubi. Sorry, I didn't delete that. I'm gonna go into this profile of some different strains in a minute. These are different haplotypes. So haplotypes, they're given, the mice, they're given letters like this. BALB-C are the D haplotype, the B6 are B. If you mate, you can mate B with D, and the progeny will be heterozygous and will express both. The MHC genes are codominant. There's different strains may differ at other loci than H2 than the MHC. It's just because they're used a lot for that doesn't mean there aren't some other differences. Sometimes one wants to put a gene from one strain onto a different background. 20 crosses is done sometimes as a number that can be done. So there are people, any research facility, big time research facility has a mouse facility and people dedicated to working with mice who know what they're doing and do this kind of stuff. And you can do this, and you can do, they call speed congenics, when you can do, you can test litter mates for the appropriate, passing on the appropriate allele. First, compared to this stuff with people, you can do population genetics, you can do a sampling of people, but you're gonna have, even if you have people of one ethnic group, you're still gonna have, some pretty good diversity of MHC or HLA. People do think, scientists do population genetics and things like genome-wide association studies with humans. Okay, let's look at this website with the, so JAX stands for Jackson Labs. So these guys. These guys propagate these mice. They also do basic research. You can buy mice from them, and they create new mice too. So let's do, why this particular, so the story, the history of this mouse. Here is here's this mouse. It's first bred by the Jackson Labs, C.C. Little, okay? And I guess it tells you an associate there. She calls this the mouse of choice. It's a good breeder and long-lived. Low susceptibility to tumors, few unfavorable phenotypes. So there are people who really know this stuff and they work with these mice all the time. But they're black and they're small. So you get the idea of some of this stuff they did. One of the oldest inbred strains. Okay. Anyway. Let's look at another one. Another widely used mouse strain. Strains do have slight differences. They might be more, some might be more prone to autoimmune disease than others. strain highlights. Well known for the production of plasmacytomas. Okay. Anyway, you get the idea. We should maintain the Colesberg hybrid. Those are a couple famous strains. So get the idea how this works. Okay. So, humans and mice are different, and the kinds of experiments we do with humans and mice are different. Basically, a lot of the immunology that we know is the same. There are some minor differences known. There are probably other things that maybe aren't fully developed. So recognize that mice are thought to be a good model for our immune system. It certainly have informed what we know so far, but there's going to be some differences. Certainly there are differences in how the experiments we do. Okay? Questions about that? And I recognize that some of you are working or have worked with mice and they know more about them than I do. Okay, so that's the first start. So I prepared this lecture a while back for another course, and I've updated parts of it, and I've decided that it's okay to just go through this. And some things aren't appropriate here. In that other class, we were doing a few experiments. Okay, so flow cytometry, what is it used for? Cytometry is analysis of cells, and flow allows us to interrogate a cell. That's one of those strong verbs. We interrogate a cell, find out what it's about. So you might have heard FACS, fluorescence activated cell sorting. That's a trademark term, although everyone says FACS. And I think when I originally wrote this, there were still FACS machines around, and there aren't anymore, or they may be around, but they're not used. So that was a little joke in there. So I'll go through this a little bit. So you're working with a suspension of cells from blood or from culture, T cells from blood. We want to know how many our CD4s have CD4. We've all heard, you all know, well, we talked about CD4 now as a marker on some T cells, but not all. You know, clinically, it's important to determine people with AIDS have a depressed level of CD4. So that's a really simple thing that everyone knows. Okay, it's a surface protein on T cells. Okay. So what one needs is an antibody that's specific. You know what so you guys know what antibodies are. But specificity, and you know, with something with proteins, you can make antibodies. So you have a reagent, a specific reagent for any protein, which is pretty remarkable. So and to really follow, I like to put a color on the antibody. So even if you've never done flow cytometry, you've probably seen beautiful experiments in the book, or you used fluorescence microscopy to see staining of where a particular protein is on a cell, or if a particular cell has that protein, and you see that characteristic green fluorescence that so many of us have seen. Things with green, with fluorescein. Okay, let's, I think at this point, I'll shift to this YouTube. I'm so poor on PCs now. Just right here.
There's a button towards the right that says skip ads.
To the right?
On the video itself.
Ritter.
Oh, I know the full screen, but how about for sound?
Sounds fine from here.
Oh, I just need to.
How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample. We will introduce you to the flow cytometry experimental technique. Flow cytometry is used to count and analyze the size, shape, and properties of individual cells within a heterogeneous population of cells. Flow cytometry data is extremely quantitative and can be analyzed in depth by specific flow cytometry software programs. Once a suspension of individual cells has been prepared for flow cytometry analysis from cells, tissues, or organisms, the prepared sample is placed on the flow cytometer. How does a flow cytometer analyze the size, shape, and properties of individual cells? A flow cytometer contains several key components, including the sample, fluidics that move the sample into the flow cytometer, lasers, optics which gather the light, detectors to sense the light, and a computer system to output the data into a form that can be analyzed by the researcher. Once a sample has been placed in the flow cytometer, the machine will suck up the sample, mix the sample into a stream of saline solution in the cytometer, and lead the cell suspension through a narrowing channel, causing the cells to form a single file line before the cells pass through the laser at the interrogation point. Each cell within the sample then moves across the laser beam, allowing each cell within the sample to be individually analyzed. As each cell passes through the laser beam will scatter in multiple directions. The flow cytometer detects light scattered in a forward manner, called forward scatter, and light scattered in a sideways manner, called side scatter. The amount of forward scattered light for each cell is detected by a detector on the far side of the cell from the laser. Forward scatter is proportional to the size of the cell. The detector converts the scattered light into a voltage pulse, which is directly proportional to the amount of forward scattered light. The computer converts these data into a histogram plot with the amount of forward scattered light on the x-axis and the number of cells on the y-axis. The amount of side scattered light is detected by a detector located perpendicular to the path of the laser beam. Side scatter is proportional to the shape and internal complexity of a cell. Just like with forward scattered light, the flow cytometer converts the detected side scattered light into a voltage pulse, which is directly proportional to the amount of side scattered light. By analyzing the forward and side scatter data together, the researcher can understand a cell's size, shape, and complexity. In addition, the analysis of the forward and side scatter data can allow the researcher to divide the heterogeneous population of cells into individual populations with varying size, shape, and complexity. This capability of analyzing multiple populations within a sample is the immense strength of the flow cytometry experimental technique. In addition to analyzing a cell's size, shape, and complexity, a flow cytometer can also detect emitted light from excited fluorescent molecules, such as fluorescently labeled antibodies or fluorescent dyes or stains. For the rest of this animation, we will focus on the detection of fluorescent signal by the flow cytometer. For more information about fluorescence, please see the fluorescence animation. The fluorescent molecules, or fluorophores, are excited by the correct wavelength of laser light when the cell passes through the laser beam. After excitation, the light emitted by the fluorophore is directed along a path with emission filters that allow for the detection of multiple different fluorophores emitting light in a cell. A flow cytometer can detect the presence of a fluorophore and also quantify the relative amount of a fluorophore within a cell. A flow cytometer can also be used to detect light emitted from multiple fluorophores in the same sample. While it is common for flow cytometers to detect 2, 3, or 4 colors at the same time, some flow cytometers can even detect up to 18 different colors at the same time. The data can then be presented to the researcher in the form of a histogram or a dot plot. Such data furthers the powers of flow cytometry analysis by enabling the researcher to analyze individual populations of cells within a heterogeneous mix of cells in the sample.
Can't go? Do this to clear out stuff.
Okay. Oh, yeah. I think that came up before. Yeah. Amusing things here, huh? So, so one thing one point I want to make about flow cytometry is that you get information about each individual cell. You can have millions of cells and you get information about each one, and that's incredible. So let's say we have some, well, let's see, I think I had something here. No, I think it's later. Okay, let's say you're interested in fluorescence, and I'll give an example. Cells expressing, I guess it can be just stained with an antibody. So let's say you have some bulk technique where you look at a lot of cells, like say and you can look at their fluorescence. So you can spin them down and use some technique to determine how much fluorescence there is there. But notice that here's the and we get some idea of total fluorescence from a whole cell population. But notice that these distributions of fluorescence I draw poorly, but you get the idea. These distributions would all look the same if you use some bulk method, like spinning down cells and looking at how much fluorescence is there. Okay, so here we have a uniform population with an intermediate value. Here we have two populations of cells, one very low, one very high, or maybe it's a zero. And then this is meant to be, I guess you can't really tell, but it's meant to be a moderate throughout. These are all going to give the same kind of fluorescence per cell number. So this is the beauty, the value of flow cytometry, that you get information from every cell. Further, let's say, jump ahead, if we're looking at two different markers on the surface and two different fluorochromes, we can see a total amount of how many are positive for each, but we can even see down the individual cell how many are positive for both of the fluorochromes, how many are positive for one but not the other, how many are the reciprocal, and how many are negative for both. Okay, so tremendously valuable, and being able to determine that down to the level of each. You get data on each cell. You interrogate each cell. Whenever I hear interrogate, it sounds like a cop show where they have the strong light on the suspect undergoing interrogation. We had color on the antibody, something like fluorescein. Use first, the reagent is FITC for fluorescein isothiocyanate. That's really easy to do. It's a simple reaction. I've done it in the lab. And you add the small molecule and then put it over a column that'll separate small things from big things. Easy to do. Okay, so. Fluorescence is great sensitivity. Incubate with our green antibody. The facts. Fine stream I was amused, you probably heard me chuckle when they said you suck up this. So I've always thought when I lecture, I've always said suck up, and I feel it's not very scientific to say that. But aspirate is the other one you can say. But anyway, so I laugh at myself for saying that, but then in the video they said suck up, so okay. So drawing up cells in a very fine stream, has in front of laser light, and it's light. Okay, so a very large number of cells can be evaluated in a second. Data stored on a file. Other measurements can be taken as well. All right, let me see. Okay, and this was a point I was making earlier. We have to make sure that our antibody is good with high affinity, make sure it's specific. So we'll see the value here. We could determine that a patient had the normal number of CD4 plus T cells, which would be about 2/3 of the T cells. So there's a lot of engineering that goes into this. Fluidics, optics, and computer power are going to make this work. So I like this analogy that, you know, car, for driving a car, like I used to have a Toyota Corolla. It's kind of pretty basic, always works, lasts forever, until someone rear-ended me on the freeway. But the idea is a Toyota Corolla will get you where you want to go. It's not flashy. Anyone, you can drive it without knowing a lot about all the engine, you know, everything it does. Okay, there's also sports cars that people work on constantly, and you know, there's kind of people that just love to work on engines, and they're always working on it. And some of those also, those high performance engines, they're prone to having problems and breaking down and needing adjustment. So that's my analogy between cars and flow cytometers. So they're flow cytometers that pretty much anyone can use, pretty, you know, with just a little bit of training. You make sure there's the right fluids in there, know how to turn it on, and it's pretty easy. There are also flow cytometers that really require more expertise. So if you're at a major institution where they might have multiple ones of these, and it is likely that they wouldn't let you use the fancier ones yourself. You would have to submit your samples and maybe be with the person that was using them, or maybe they let you after a while. But others, the simpler ones are pretty easy to work with yourself. So that's my analogy. Okay. So as mentioned in the video, measurements are taken for forward scatter and side scatter. It's actually a small forward angle scatter, and it's roughly related to the size of the cell. The side scatter is at 90 degrees and is related to the complexity. That's relevant to us because granulocytes, which we mostly won't talk about here, have a high side scatter, and lymphocytes are distinguished from them by their lack of high side scatter. So for these parameters, the important thing is not the absolute measure, but the pattern. So we also use forward scatter and side scatter to distinguish live cells from dead cells and debris. It says here, these two areas, immunology and flow cytometry, have grown up together. This goes back to, there's a couple of the Herzenbergs at Stanford who really developed this. So single cell suspension to analyze our cells. So the immune system has benefited a lot from flow cytometry. Also, immune cells are ideal for this because you do have individual cells that, you know, some of them tend to stick together a little bit, but basically won't too much. But think about cells that are part of a tissue where they stick together. You have to disaggregate them, and you might be changing them to do that. So, but the immune system cells, or cells in culture, are ideal for this kind of work. So we see how these two areas have really grown up together. But other things can be run on a cytometer as well. Okay, now, more interesting, we can use multiple antibodies if each has its own color, because we can detect different colors. You can even look at proteins inside the cell by doing intracellular staining. And with some machines, you can not only analyze your sample, you can actually sort, so that you will sort for only the cells that are positive for a given marker, and then do a sterile sort with the whole system, fluidic, sterile, so you can purify those cells. So that's pretty incredible. So you could sort T or B cells from mice with a particular pathogen and then inject them into a different mouse. This is a particular machine that I used at that point. So I say a very open-ended field. There are always newer, faster machines that can do more, and I find it pretty incredible. So this is an experiment I was going to do with this other class. Antibodies, sometimes people have to make their own antibodies for a new protein, but for the common things that are widely used, companies make them, and they're very profitable once they have them, and you can buy antibodies conjugated with different colors for all your favorite immune system surface markers. Kinds of numbers, often 10 to 20 million cells per mil, often stain in these kinds of volumes. You may need to use fewer cells. People stain on ice. We say react with the antibody. Cells survive better on ice. They won't cap or cluster. They can come together. And they won't take up antibody. Often stain for short periods of time. So a little bit about software. So we set gains to define populations, cells you want. So often an early step is to look at forward scatter versus side scatter. And there are gains that you set, or voltage you set, and you want to set these so that your cells are away from the axes. So here's a typical sort of pattern that you might not, oh, I got it. Yeah, I think I have another slide of that. Another. So here is a histogram with forward versus side scatter, and people have defined a region here, R1. So lymphocytes don't have the amount of side scatter that granulocytes have, so these things up here are likely granulocytes. These are likely lymphocytes. Gain is adjusted so that you don't want any population to be pressed against the bottom or either axis. Something you notice here is that there's nothing below 50. There's a couple of things here, and I don't understand that. Sometimes one thing people do is set a threshold, which means the machine will only record things bigger than a certain size. This eliminates dust and stuff. So you want all the information, but you arbitrarily pick a size that things smaller than that. We're going to assume it's dust, it's debris, not whole cells. So that's called a threshold. And then often people look at forward versus side scatter, and we might, for instance, This is likely a experiment designed to look at lymphocytes, and this region is picked here, and one draws a line around that for further analysis. And you can draw that, and you can change that later, unless you're doing a sort, and then you've already committed to it. Okay, a little bit about that.
A question.
Yes.
So you're isolating this because they are clumping together?
No, because they're not clumping.
Yeah.
They're not clumping together. They are clustered together in your analysis.
Yeah, not physically.
Yeah, not
but-
physically clumping.
okay.
Yes. Yeah. So you try to. So there's some subjectivity in this. Where you draw the line exactly, but that's really cool. Okay. So those are early steps here, and just looking at the cells that we want. Lost my. There's a threshold function, what I mentioned, so example of a region. So now we also adjust the gains for the appropriate fluorescence channels, so the negative control is in the first decade, but not pressed up against the axis. So two negative controls. One is unstained cells, and also an isotype control, meaning an irrelevant antibody of the same type. What does this guard against? It guards against non-specific staking. Or where the antibodies come from a company. Most today are monoclonals. Antibodies ideally are specific, might be some cross-reactivity. Okay, yes?
So the negative controls are the unstained cells?
Unstained cells, and also different controls. Unstained cells, there could be autofluorescence, something that's in there that fluoresces even without you're adding an antibody, or also an isotype control.
What is the meaning of isotype control?
What?
What does isotype control mean?
Oh, okay, sorry. So antibodies have a particular constant region, and that's said to be the isotype. So another IgG2A an irrelevant IgG2A would be an isotype control if your experimental antibody is IgG2A. Okay, let's see. Can we compare two results with the same antibody and same fluorochrome? Yes. If we stain cells with a PE, that's a red color labeled antibody, and stain the same cells with the same antibody labeled with a FITC. If we compare levels of surface expression, no, because there might be more PEs on the antibody than FITCs. Where one molecule of PE is brighter than one molecule of FITC. Quantitation is possible by using spheres with a known number of fluorochromes on them. Let's see. After setting a region, you can check to see if your data back up your point you were trying to make. You must play the role of devil's advocate for this. Okay, so I think I'm not going to go to this in too great detail, but you might find, if you find that a minority, if you're looking for make a certain point, and I made up a marker 1, 2, 3, 4, and if you define a region and find that a tiny number of your cells express this marker, then you might go back and see where they are on the forward versus side scatter, and maybe if they're all, if you might look at it again, decide, well, maybe you drew the forward versus side wrong, and those aren't really the cells you're looking for. Okay, so be the devil's advocate if you're trying to prove a point. If the cells that express the marker are clustered near the debris, that's suspicious. It might be that only the smallest cells express the marker, but you might want to verify using something else. Okay, indirect staining. So we can use an antibody and then an antibody against an antibody. For instance, a mouse antibody and then a Go anti-mouse antibody. Show it here. You do this if you don't have a labeled primary antibody. You could use one secondary for many experiments. This can amplify a signal. Secondary antibodies are usually polyclonal and are made in a large animal like a goat or a donkey or a sheep. It's important to use proper controls. Another example of indirect staining. I've mentioned all antibodies so far. Do you have a question? You probably know about the interaction between avidin and biotin. It's one of the strongest interactions in biological systems, so that can be used in the same way that antibodies are used. If you tag a protein with biotin, it can then be stained with streptavidin as like an antibody. So, for instance, putting biotin on the antibody and then using streptavidin, and I have that for streptomyces with a fluorochrome. So it's much like using a secondary antibody. Okay, little bits here. Some people stain with only the FAB fragments to prevent cross-linking of receptors and activation of cells. Sometimes you want to avoid that if you want to, and when antibodies can cross-link surface proteins, and that can lead to an activation of cells. So here's something that's a good technical tip. So, you know about the Fc portion of antibodies, right? And monocytes, B cells, other cells may contain Fc receptors. That's what they do. They bind to the Fc portion of antibodies. This could be a problem, right? So this is a big concern if you're using mouse cells with mouse antibodies. So often people, when you're staining cells, you incubate, you spin cells down, incubate them, you often have a small amount of serum present. And the serum has a wide variety of antibodies, and this can block this non-specific staining. But if you're not using serum to block this non-specific staining, then you have to worry about that. Okay. Staining whole blood is not a problem due to the presence of Ig. Okay. Forget what this link is. Let us see. Okay, here's a whole bunch of fluorochromes. A lot of stuff here. Now, let me look at some other things. So now, let's get interesting. I don't want to take too much more of your time because I realize it's hard to listen to anyone for much more than an hour. Problems arise. Well, the great stuff is when you can use more than one color, more than one antibody, but also can lead to problems. Here are some widely used colors. FITC, green fluorescence. PE is phycoerythrin. Allophycocyanin is APC per CP. So, just look at these two for a minute. So, you can set appropriate filters. You have your wavelength for excitation that you can use, and you can set filters to only detect where you want to detect. But there are some problems. Some of the FITC here bleeds into the PE signal. Okay? And usually it's more from the lower wavelength, higher energy bleeding into the others. So when you do an analysis with multiple colors, then you have to worry that something you detected in this area here, it's going to be contributed to by PE, but also by FITC. Now, this is drawn like it's very little, but what if there were a lot of the protein that's conjugated to FTSE on the cells and not many the PE? So this could be much more of a problem. So people do this by solving, this is essentially a series of simultaneous equations. Bring back math for people, hope that's not an unpleasant experience, but that's what, this is the basic problem, overlapping spectra, and how do you account for that? And here are a bunch of different spectra, some of the same ones we've seen here. And so you can figure out all this information. Oh, the filters with the Toyota Corolla spectrophotometers, the simple ones, the windows are fixed. And then with other machines, you might be able to put in different filters and adjust that. So you get an idea here. So here's a discussion of compensation. How do you determine compensation? So originally, this was done manually by a person just looking at the data and then adjusting the gains. And I'll show you how that's, this is a whole chapter on it. Believe me, there are people who are really into this. So here we see this data here, FTSE, CD3 on the x-axis, and we see PE on the y-axis. So we want to, this is a staining with no PE added, with FTSE on CD3. But we see here these cells have not been stained with any PE antibody. There's only FTSE antibody. But because of that phenomenon we talked about before, spillover, you can see the y-axis, this is fairly high. So this is, well, it's higher than it should be. So this is our control, our unstained control. And then we see this signal is due to spillover. So the researcher sets the gain for that signal and essentially subtract a fraction of the FITC that spills over into that spills over into the PE. Okay, and the way you do that is adjust it until the center of this smear is about equal to the center of this smear. Okay, so sorry, here's uncompensated. I should have said this first. This is undercompensated, not enough. This is proper compensation. This is overcompensation. Okay, and this is done. Formerly, this was always done just by eye, and now they have a lot fancier ways of doing it. You might be looking at all the data. Okay, does that make sense? So that's very generally how things are done. So let's apply compensation if there's any kind of spillover. And let's see. I think that's all I wanted to show from this log. And here's this idea here about the spillover in essence, compensation. Okay, so now imagine, so when I used to do experiments, I work with 3 or 4 colors. Oh, often, it's common too for sometimes to use something like prapidium iodide, which will stain which will emit at a really high wavelength that will stain dead cells, so you can gate them away. That's another one. So I used to experiments with 3 or 4, and now imagine in recent years now I hear that you can stain with like 18 or 20 fluorochromes, and it's mind-boggling. And I really couldn't believe how they could do this, but essentially, as discussed by, let's see, did I look at this one yet? That's the one I looked at. So this is called spectral cytometry, and I'm still not quite sure I believe it, but people do this, and they can look at so many different markers. So with spectral flow cytometry, allows you to use many different markers. And essentially, the short explanation seems to be that you look at the whole range of wavelengths in the area, and from knowing shapes of the curves, you can do the proper calculations to determine what part of the signal is due to the fluorochrome you want and what is not. So, spectrum 40 plus colors, sure.
Welcome to the Intro to Spectral Flow Cytometry educational series. My name is Mirko Bersevi. In the next few videos, we will go through some of the fundamentals of spectral flow cytometry, what it is, how similar and how different it is from conventional flow cytometry, before diving deeper into some of the unique features of spectral flow cytometry, such as autofluorescence and mixing, fluorochrome choice flexibility, and increased panel size. But let's start from my practical examples. If we take 2 primary colors like blue and yellow, we mix them together, we can obtain, for example, green. But we also know that if we have different contribution of each one of these primary colors, the result will be different. Now, by virtue of knowing the primary color that we use, we can guess or infer what was the relative contribution of yellow in this case from the green or the cyan color. Now, when we are looking at flow cytometry, we're looking at somehow a similar scenario, where we are now staining ourselves with antibodies that are bound, conjugated to fluorochromes. And each of these fluorochromes have a unique signature. You can call it like a primary color. And their intensity is a function of the abundance of the markers that we are analyzing. Now we can mix all these colors together in a multicolor panel. However, the flow cytometer will record a total emitted light that is the results of each individual contribution of the marker. But ultimately, we want to know the patterns of expression, the distribution and the abundance of each marker individually. So we need to go from the total back to the individual contribution. And that's the process that we use in flow cytometry, conventionally named compensation. And this is something that is now different in spectral flow cytometry. But fundamentally, the problem is the same, the goal is the same. What is changing in spectral flow cytometry is hardware component as well as software. So let's take a little bit deeper. In conventional flow cytometry, we know that we have one detector assigned for each fluorochrome, and the signal of each fluorochrome is captured through the use of filters that capture only a portion of the total signature for each fluorochrome. Now you can also see that in some of the detectors you might have an overlap and contribution of different fluorochromes to the total signal in this detector, and that's when we use our signal control as a reference, and then to calculate compensation and eliminate the signal coming from the other fluorochromes, and determine the exact contribution of, for example, the green dye. Now, Spectrum is trying to do exactly the same, but the first fundamental difference is the use of more detectors than fluorochrome. And most importantly, we are collecting, in this case, through a series of filters and detectors, the entire spectrum of emitted light, not just a portion. We still need to define the individual contribution of each fluorochrome, and we do this through two different calculations, two different mathematics. We have compensation in convention and spectral mixing in spectrum. But again, fundamentally, we are trying to define the contribution of each fluorochrome to the total fluorescence that is measured by the flow cytometry. But the fact of collecting the full spectrum, as we will see, provide additional advantages and features that are not available in conventional flow cytometry, and we'll describe in this series. But let's look first at how a conventional flow cytometer look like and how we can have a spectral flow cytometry instead. On the left, you see here a BDFX Symphony A5 cell analyzer. This is a conventional flow cytometer. It has 5 lasers. It has 28 detectors. And by definition of conventional flow cytometry, it means that we can analyze up to 28 fluorochromes. And then for each fluorochrome, we have specific filters that will help collecting the lights in these detectors. But what you can see again, that there are gaps. There are areas of the spectrum that we are not measuring, that we are ignoring. So in order to have a spectral version of this instrument, what we did, we filled those gaps with additional detectors and filters. So now we can cover the full spectrum. We have now a total of 48 detectors, and this is a truly spectral machine because we have coverage of the full spectrum, either by filling the gap with individual filters or by also splicing up the spectrum here with multiple filters. This is another view of the same concept where the light is collected in different detectors. We get a signature across each laser, and then what we can do, we can stitch it all together, and that will give us what is now very commonly represented and used as a spectral signature, where we're seeing now not just the signal in a specific and discrete portion of the spectrum, but across the full spectrum.
I think we're going to stop it there. It's really kind of getting into the weeds there, but get the idea. But I find it amazing what they can do now. I'm still sort of a little bit in disbelief, but so you can now use these many colors with many antibodies. I don't have a good feel for, if you really want to prove something, and I wonder if, and you're saying you think you found something that's contrary to the prevailing wisdom. I wonder if you don't go back and do it with only a couple fluorochromes just to be absolutely certain. But it's still remarkable what they do. And I think you have a little bit of an idea. I mean, the main thing to know is about compensation, why you need compensation for conventional, and be aware that you can use the spectral flow cytometry. Okay, so I hope that was a good overview of flow cytometry and a little bit about working with the mice. Another point on the flow cytometry, I wasn't aware for a long time that I used to think that the science, you were either professor at Harvard with great resources and you could do all sorts of things, or you had to be someone else was your boss and you could do what they said all the time. But there's a lot of things more in between now, and the people who work at the flow cytometry facility at Fred Hutch or UW or equivalent place, they have some interesting jobs, and I assure you that the people who are in charge of those facilities are really respected and are really important parts as well as they have a number of people working under them. So I know that was one of the core things that people had the opportunity to go to at Fred Hutch. You guys are a little beyond that, right? The people who were doing internships at the Corps. No one's looking like they did it, but those kinds of things are more. There are a lot of jobs there, and some of them are, fairly interesting too. So, anyway, just a thought. Okay, so there's a little aside, but this will inform everything for the future. You're talking about working with how we work with mice, and we'll be seeing a lot of flow cytometry. Okay. So you guys want to?